Hello, everyone. Good afternoon. Thank you very much for joining us for the Ensembl browser workshop today. To introduce myself again, my name is Ben Moore. I'm one of the members of the Ensembl outreach team, and I'm joined by my colleague today, Louise. Together, we're going to be teaching you about the Ensembl genome browser. I'm going to hand straight over to Louise. Louise is going to be taking you through most of the material today. But I'll be on hand to answer questions in the chat box and during the exercise sessions as well. So yeah, handing over to Louise.
Thanks, Ben. Hi, everyone, and thank you for coming today. My name is Louise Mirabueno, and I am here with Ben, who's just introduced himself. We are part of the Ensembl outreach team, and we'll be delivering today's and tomorrow's workshop. So this workshop will be recorded, and it will be shared with you once it's ready to be shared. Okay, so this workshop consists of several modules, and each module will be following this structure. We will start with a presentation where we explain what the tool is or what data is presented and how it is produced and processed. Then we'll run through a demonstration where we will show you how to get this data on Ensembl. And then at the end, you will have some time to do a few exercises. All the training material will be available on training.ensembl.org. There you will find the presentation, walkthroughs, and exercises, and a living document. So what I'm going to do now, I'm just going to show you this website where you can find all the materials. So I'm going to open a new tab and type in training.ensembl.org. And this leads you to the Ensembl training page. So if you click down here on the left bottom, there it says find materials from training. So if you click the link there, and our workshop will be the only workshop that is on the page. So click on that link as well. And here you will find all sorts of material that we will be covering today and tomorrow. So we've got the a link to the presentation here on the right-hand side. We've got a link to the living document, which I'm going to open now. This opens a new tab, and it is just a document with general information such as the schedule of today and tomorrow, links to some resources, and there's also some space for questions. So if you have questions, you can either unmute yourself, you can type in your question in the Zoom chat box, or you can go to the living document to the bottom of the page and type in longer questions, for example, there. It's just loading right now. It's a bit slow, sorry about this, but I will scroll down to the bottom just to show you how the question section looks like. Here it is. Okay, so the question section is on page 4. So all you'll need to do is type in your question, and then we will answer it as soon as we see it. You can write down your name, or you can also ask anonymously, whichever you prefer. And then further up of the document, we've got the resources I mentioned, and some golden rules for efficient online training, and the workshop agenda for today and tomorrow. So I'm gonna close this for now and go back to the training page. And one more thing I want to mention are the demos and the exercises. So you find the demos and the exercises here at the bottom of the page, and they are all categorized in tabs. So according to what module I'll be covering, you can try answering the exercises here. There are several exercises per module. So if you're, for example, interested in mosquito, feel free to only answer the mosquito question. There are text answers here, so I'm not going to click that because it'll show up. But if you're stuck, you can look at the text answer or the video answer, or you can also just ask us on Zoom or the living document. And if you want to go through the demonstration, which I will do after each module, here is another tab that that's just pictures of the demonstration that I will be doing, and Ben as well. So it's just for if you didn't follow along, you can always come back here and just review what I've gone through. And this training page will be available indefinitely, so you can use it as a reference in future for your work. Okay, so I'm going to go back to my presentation now. So I've shown you the training materials, and now I'll just go through the different modules we'll be covering. So today I'll be covering an introduction to Ensembl, show you the region in detail view, and talk about genes and transcripts. And at the end of the day, Ben will be covering the comparative genomics module. And tomorrow we will come back at 1 p.m. and cover the variation module, regulation on Ensembl, and also Biomart, which is a tool on Ensembl where you can extract a large amount of data from the website in one go. And we will finish up with some closing remarks as well. The objectives of this workshop is to introduce you to the Ensembl genome browser, what type of data you can get in Ensembl, and how to navigate the website and where to find help and documentation. So with that, let's start with the first module, which is an introduction to Ensembl. Before I continue, does anyone have any questions? Wonderful. Okay, so let's start with the first module. I will cover why genome browsers are needed, what Ensembl is, and what data it offers, how genome assemblies are made, and then I will give a brief demonstration and you will have some time to do some exercises. So I'll start off with a bit of background. In 1977, Fred Sanger and his team sequenced the first genome, which was a bacteriophage, paving the way for sequencing genomes of much more complex organisms. The first draft of the human genome was published in 2004, a project which was started in 1990. And since then, many, many more organisms have been sequenced. And today we can sequence a complete human genome in a matter of days. And Ensembl was launched in 1999 with the goal to automatically annotate this first draft of the human genome and integrate this with other data and make it publicly available. Why do we need genome browsers like Ensembl? With sequencing being more cost-effective and faster, genomic data is piling up quickly. But when sequencing data is produced, you get something like this, which is just a bunch of letters without a meaning. And trying to read a genome without any annotation data would be like trying to read a foreign language without a dictionary. So it won't mean anything to you. And this is what genome browsers are there for. And Ensembl is a type of genome browser. You can use genome browsers to map annotations to genomic data. And Ensembl brings together information from a wide range of other databases in a single site. You might have heard of other genome browsers, for example, UCSC and NCBI. And I also just want to note that Ensembl does not do any of its own sequencing. We simply take publicly available data, sequencing data, and use our own pipelines to annotate the genomic data. There are many features that are annotated by Ensembl, which include genes, variants, promoters, binding sites, and loads more. Genomes that are annotated with genes are referred to as gene builds. So I'll be using, you'll see that word quite a lot on Ensembl, so just remember that. And we also have variation data in a number of different species, comparative genomics data such as alignments, gene trees, and homologs. And there is also some regulatory builds available for human and mouse from experimental data. And you can also use a number of tools on Ensembl to export, for example, large amounts of data, which is Biomart, a module we'll be covering tomorrow. And you can process your own data with the variant effect predictor, which Ben will be covering tomorrow as well. Or you can access the Ensembl database via APIs. And Ensembl data is completely open source, and whole genome files can be accessed through the FTP sites, which I will show you during the demo. The Ensembl browser is dedicated to vertebrate species only, but as we know, vertebrate species are only a fraction of all the organisms in the world. However, you can find some non-vertebrate species, for example, fly, worm, and yeast genomes, because they're commonly used model organisms. If you're interested in non-vertebrate species, you'll have to visit the Ensembl sister site, which is called Ensembl Genomes. And this is divided into different taxa. So we've got sites for plants, metazoa, bacteria, protists, fungi, and also a site for the COVID-19 virus. So once you are familiar with the Ensembl browser, the Ensembl Genomes browsers are all structured in the same way. So once you're familiar with the Ensembl browser, you'll have no problem navigating through all the other browsers as well. Because genomic data is constantly evolving and changing, Ensembl is updated regularly every 3 months to represent the most recent findings. Behind every update, a lot of work is involved, which includes the addition of new genome assemblies, underlying software updates, updating gene sets, addition of new comparative genomics data, updated variation and regulation data, and just in general, new or updated interfaces. The last release was Ensembl 105, which was published in December 2021. For Ensembl Genomes, it was release 52. And just a couple of weeks ago, Ensembl 106 or Ensembl Genomes 53 was released. And because Ensembl is not static, you need to make sure to write down what Ensembl version you are working with. If you're working with an older version, we keep Ensembl archives for up to 5 years. So you can still use the, for example, Ensembl 105 browser for your research. I'll show you how to find the archives and where you can find what release you are currently working with during the demonstration. So new genome assemblies are published all the time, and therefore, apart from Ensembl and Ensembl Genomes, we also have an Ensembl rapid release browser, which is released every 2 weeks. And this browser includes genomes with gene annotation. And the only other tool that you can use is BLAST. There is some comparative genomics data available, which are gradually being released, but there is no biomart or variation data. Okay, so let's look at genome assemblies. The human reference genome is not a genome of a single person, and it is not a consensus genome either. It's made up of fragments from different individuals, and each fragment represents a real person's genome, making the reference genome some sort of mosaic or hybrid genome of different individuals' DNA, rather than a consensus genome, which would be made up of the most common sequences found in the pool of samples that were sequenced. And note that genome assemblies are not a perfect representation of the genome. There are still gaps in genome assemblies, but researchers fill these gaps and correct errors and share patches continuously. This means that there can be different versions of a genome assembly. So for human, Ensembl offers 3 versions, which is the GRCH38. And this is the most recent and also the most supported human genome assembly. It's got the fewest gaps, and many rare alleles have been replaced with more common ones. We also have a dedicated site for the GRCH37 assembly, as it's still widely used in research. But this assembly has some large gaps, and there are limited updates for the data available. And finally, we also have a dedicated site for the NCBI36 genome assembly, but this has even more gaps than GRCH37, and it is no longer being updated. So what can you use Ensembl for? To summarize, you can explore genomic regions in the browser, learn more about the genes and variants of interest, and find out more about the genetic background of a phenotype. So with this, I'm finishing the presentation, and I will move on to a demonstration where I will show you how to navigate the browser and where to find information about a species and a genome assembly. So what I will do is I'll open a new tab in my browser, and I'll type in ensembl.org. So this brings us straight to the Ensembl homepage. And the first thing that you'll see is the top page, the navigation bar. We've got the Ensembl logo here. So if you click on that, you will be sent straight back to the homepage, just like any other website. We've got links to some tools, so a BLAST, VAT, and also some other tools here. We've got a link to Biomart, downloads if you're looking for help and documentation, and also a link to our blog where we release updates on Ensembl. So if you wanted to get whole genome, if you wanted to download whole genome sequences, you'll have to go to downloads, which I will click now. And here on the left-hand side, there are different sections, and you can click the FTP download. And this opens up a new page with all the different species that you can download. So the whole genome data files. You can, so the species you can find here in this table. And if you know what you're looking for, you can just type it in here, for example, human, and it will filter the results. Okay, so I will go back to the home page by clicking the top left logo. And I'll show you the rest of the homepage. So here on the right, we've got a description of Ensembl. We've got the latest updates in this release. We've got a link to the rapid release site, which is updated every 2 weeks. And also some blog posts from our blog. If you go further down, you can see a link on the right-hand side on the bottom that says View in Archive Site. So if you click that, it opens up a pop-up window, and these are all the archives that you can visit. And archives are saved for up to 5 years. To get out, you can click this check on the top right, or you can just click an empty space outside of the pop-up window. If you wanted to share a particular page on Ensembl with your colleagues, you can click the permanent link button here on the right-hand side. And that'll just give you a static link for the current release that you're on. And then at the bottom of the page, we have some more links. So earlier I mentioned our sister site, Ensembl Genomes. You can go straight to those sites by clicking these shortcuts here, or you can open a new tab and type in ensemblgenomes.org. So this brings us to sort of a landing page where you can choose which Ensembl genome site you're interested in. So we've got plants, metazoa, protists, fungi, and bacteria. And to visit a site, you can just either click on the button down here, which says go to Ensembl bacteria, or you can go to the page directly by typing in, for example, bacteria.ensembl.org. They will both send you to the same page. And as you can see from the Ensembl homepage, it is structured just the same way as Ensembl. So I will close this. And now I'm back on the Ensembl homepage. I'll scroll up to the top. And I want to show you the different search bars that we have. So we've got a general search bar here on the top right-hand side, where you can just type in, for example, gene, and this will search this gene in all of the species in Ensembl. But if you know what species you're interested in, you can just type it into this search bar here in the middle of the page, and that will just search it in the species that you select from this drop-down menu. Further down, you can see that you can select favorite genomes as well. You can edit this and add your favorite genomes to it. But when you do that, I just click the edit symbol there. When you do that, you'll have to log in. So registration is free. You can delete it at any time, and it's really helpful for saving favorites, or for example, later when I show you, you can save several displays, tracks, but this is optional. So if you wanted a bit more functionality on Ensembl, you can register and you'll have access to these modifications. So here on the left, we've got another drop-down menu. So when you select a species here, it will send you straight to the species page. But before I do that, I just want to show you the view full list of species link. So I'm going to click this, which is here at the bottom left. and it opens up a table with all the species that are available on Ensembl. So we've got the table has got several columns, and we've got the common name of the species, scientific name, taxon ID, the assembly that is available at Ensembl, accession number, and the gene build method, so how it was annotated. You can choose to hide or show columns by clicking this option in the middle of the table. And you can also filter the table by typing in, by typing in your species of interest in the search box here on the right. So for example, if you're looking for human and you don't want to scroll down, just type in human. So it's taking a little while to load. I can see that the page is still loading here on the top, on the tab. So I'm just going to let that update for a little while. Does anyone have any questions so far? Cool. Okay, so this is taking a little longer than I'd like. So what I'm going to do is I'm going to open a new tab and go back to the Ensembl homepage. Can I just check with you guys? Is your Ensembl being slow as well? So if you just open the page.
Yeah, it's slow.
It's slow as well for you. Okay. I was hoping it was just my internet. Okay, well, that's going to load for a little while now. But what I wanted to show you is you could filter this list of species by the name by the species you're looking for. So if you're only interested in mouse species, you can just type in mouse. I can see that the other tab has loaded, so I'm going to change tabs. I have a feeling the species list tab is being quite slow because it's quite a big table. So I'm gonna get to the species page in a different way. So what we did there is click on the full list of all species link, but instead what I'm gonna do is I'm gonna actually click on human right here, which will do the exact same thing as clicking the species link in the species list. Okay, so this is the species page, and you can find all sorts of information on human in this page. So we've got another search bar that is species specific. So if you look for, if you're interested in a gene, you can type it in here, and when you click go, it only searches the gene in human. So what else we got? So we've got genome assembly information here. You can choose to view more information statistics, which I will click on in a minute. You can download the sequence, convert coordinates, display your own data on Ensembl, but you can also view other assemblies of the human genome. So if you remember from earlier, we've got 3 different genome assemblies of human. So we are currently viewing the GRCH assembly, which you can see here on the top left. But if you wanted to use one of the other assemblies, you can click this drop-down menu under other assemblies. And you'll have the option to select GRCH37 or NCBI36. If you click that and then click go, it'll open a new page, which will send you to the assembly that you selected. So here on the right, we've also got gene annotation information, so you can find out how this gene build was made. You can download FASTA files of different sequences or download annotation files. And we've also got comparative genomics data, regulation data, and variation data. So these are modules that Ben will be covering with you today, and regulation and variation tomorrow. So I want to show you where you can find more information about genome assembly. So I'm going to scroll back up and go to this genome assembly section here on the left. And if you click more information and statistics, it will open a new page. And this is just giving you information about the assembly that you selected and some summary statistics. So here you can read where the assembly was from, some patches that were made. You can select other assemblies just like in the site before. And you can also find more information on the gene annotation of this assembly. On the right here, you can find the summary statistics, so you can see where the assembly was originally submitted to, the length of the assembly. We've got the assembly provider, annotation provider. So in this case, it was Ensembl who did the annotation for human in this genome browser. And you can also see some others summary, for example, gene counts, so the number of coding genes, number of gene transcripts, and so on. So that finishes up my demonstration for this section. I will go back to the presentation, and you'll now have time to do exercises. So I'll just open up the training page again, just to show you where you can find the exercises. So we'll be working on species and genome assemblies. So back on the training page, that's just the first tab. And there are loads of exercises, so feel free to do whichever exercises you feel like doing. I will give you 15 minutes to do this, and then we'll come back to do some more, another module, sorry. So Barrett, sorry, I hope I pronounced that right. So you asked what the golden path length is. So that is, I believe you saw it here. So the golden path length is just an alternative measure of the base pair length. So an alternative measure of this. And basically it omits redundant regions like haplotypes. So in other words, it's the length, the base pair length of all the top quality sequences.
I was also going to add in a separate comment on Louise's answer as well there. So historically, the base pairs, the number for base pairs and the number for the golden path length were different depending on the species. So the base pairs would be the total number of base pairs in the genome assembly for a species. So that included the primary assembly and then all of the redundant sequences like Louise mentioned. Whereas the golden path length was the path through the genome with removing the redundant sequences. However, we got so many questions about this that it was quite confusing for users and people who were looking at that data. So what we actually have done is we've just put the same number twice here in this case. So you can see here, even though human does have redundant sequences, the base pairs and the golden path length are exactly the same. So this is the length of the human genome with all of the redundant sequences removed.
Okay, so it's 13:55 now. I can see that Daniel asked a question, but I might let Ben answer that so I can continue with the module. And before I move on to the next module, actually, I want to show you the Ensembl mirror sites. So just earlier, you could see that Ensembl can be a bit slow sometimes. So to get around that, you can visit one of our mirror sites. So I'm going to go to the Ensembl tab, and on the navigation bar on the top page, if you click on Help and Docs, you will see these different sections on the left-hand side, and quite further down under About the Ensembl project, you can see a link that says Mirror Sites. So if you click that, you can see the mirror site you are currently on. So we're on the UK one, but you can also visit the US West, US East, and Asia mirror sites. So if you click on one of them, it'll open it. And to know that you are actually on a mirror site, you'll see a little flag next to the logo on the top left-hand side there. So Ensembl is working all right at the moment. So I'm just going to go back to the usual Ensembl page and continue there. Right, back to the presentation. So we'll now look at a specific region in the human genome. And for this, we'll actually go straight to the demonstration. And in this demonstration, I will show you how you can manipulate the region in detail display and view data that you're interested in. So for that, we're looking at specific coordinates. I'm going to copy these and put them in the chat box so you can copy paste. And with that, I'm going to go back to the Ensembl browser tab. Okay, so we are interested in specific coordinates in the human genome. So what I'm going to do is I'm going to select a species in the search bar in the middle of the page from the drop down and click on human. And I'm going to paste the coordinates into the search bar. So I've already got them copied, so I'm just pasting them. The structure of the coordinates is number, which represents the chromosome, followed by a colon, and then the start position, followed by a hyphen, and the end position. So I will click go, and this will lead us to results page. Sorry, I thought it was going to send us to the results page, which would give us multiple options. But because we search for a specific coordinate, it's actually going to open the location tab straight away because we know which location we're looking at in the region. So here we are in the location tab, which you know you're at when you look underneath the navigation bar here on the left. And in this tab, we've got different displays that we can choose from. So on the left-hand side here, there are a collection of different displays. So you can choose to see the whole genome display. You can choose to view the region in detail display, which we are currently on now, or you can also choose to view variants, and there are also hyperlinks to other genome browsers such as UCSC and NCBI. So those are the displays for this location tab. Underneath, you can also see a few more other options. So you can choose to configure the page by clicking this button, which we're going to look at a bit later in this demonstration. You can choose to add tracks. So tracks are these lines here in the displays, and they just represent different elements that are annotated on the sequences. You can choose to export data. So if you click that, it will open a pop-up window, and you can select different formats that you want to download from. You can also select a different location. So if you wanted a bit of a different location, you can paste your location in here. You can choose to add flanking sequences. And when you click next, it will just open a new window showing you which format you want to download. So I'm not going to do that for now. I'm going to go out of this pop-up window, and I'm back on the region in detail view. So I've shown you the export data. You can also choose to share this page. So you can click on that, and if you wanted to share this particular view with a collaborator, clicking on the share this page button will give you a URL that will send them straight into the view you're currently seeing. And you can also choose to bookmark this page. But again, for this, you'll need to sign up, register on Ensembl. So I'll scroll back up, because now I want to show you the right-hand side of this page. And these are just 3 displays that are the same location that we're looking at, so which is this location here in this tab, but each of them is in more detail. So the first diagram we see here is the chromosome overview. So if you look at this little red box on the chromosome, that is the location that we've selected. And you can also see here again on the title, the location positions that we've entered earlier in the search box. For the chromosome diagram or chromosome view, you can see the cytogenic bands in gray and alternating gray. You can see the fixed patch. So if you hover over this track, or if you hover over any track, you'll be able to see what the track represents. And for this particular track, it represents the patches or the sequences that have been improved in this assembly for known errors. So if you look closely, there are green and red dots there, and the green dots represent the known assembly errors that were fixed or patched, and the red ones represent the ones that have not been fixed yet. Scrolling down, we see the region in detail overview, which is in a 1 megabyte view. So here again, we see a red box that highlights the position we entered, which is the same position as it was on the chromosome. And if you're unsure of anything on Ensembl, some sections have a question mark on them, and that means it will just send you straight to a documentation page which explains what the data is about. So I just click that and it opens a new tab or window. And here you can just read a bit more what the different sections are and what data you're seeing in there if you ever forget. So I will close this documentation page, sending me straight back to the region and detail view. And yeah, so as mentioned, it's the same location as on the chromosome, but it just in a bit more detail. So here you can actually see a few more tracks. So we've got the chromosome bands, which you can see here in this little on the chromosome diagram above. You can see this context path, which is just the genome assembly track. And these are in alternating blue and light blue and dark blue. And then you've also got the genes track, which just gives you the name of the genes that are located in this region. Yeah, and then also you've got this regulatory build, which Ben will talk a bit more about tomorrow, but it's the different features, the different regulatory features that are present in this current region. And the legend for that, you can see underneath. So we've got a legend for the regulation build, and we've also got a legend for the genes build there. So scrolling further down, we'll see an even more detailed view of this red box that we highlighted in our positions. So this is the most detailed view of our region. And here you can see even more tracks. So we've got the same tracks as above. We've got the context track, the genes track, but also we have more detailed information about the genes on here. So the genes, there are two genes tracks, as you can see here. So we've got the context track in the middle, but we have a genes track above and a genes track below. And that's just simply dividing up the genes that are in the forward and in the reverse strand. So genes that are located in the forward strand are above the context track here, and genes that are located on the reverse strand are located underneath the context track. And you can also see the orientation of the gene by looking at this little greater than sign. So greater than means forward strand, located on the forward strand. And then if you look underneath the context track, the less than sign here, that means the gene is located on the reverse strand. So on Ensembl, genes are actually displayed as transcripts. I should have mentioned that a bit earlier, I'm sorry. But one gene on Ensembl is a collection of alternatively spliced transcripts. So you can see here that the NUDT6 gene, it has loads of transcripts in this location. So that just means that NUDT6 transcribes into multiple transcripts. I would talk about it in more detail, but if you look at the individual transcripts here, you can see that they are made up of different symbols. So we've got a box, then we've got a line, we've got a box, line, and then box again. So the box represents an exon, and the line represents the intron of the transcript. Okay, so you can also scroll further down, and similar to the region and overview earlier, so a bit less in detail, there are legends that you can find at the bottom of the page here. Legends for the tracks. Okay, so I'm going to scroll up to the top of this diagram. And what I'm going to do is I'm going to show you how to manipulate this view. So you can click either on this cog here on the diagram on the top right-hand side, top left-hand side, or earlier I showed you the different options on the very left. So you can also click configure this page here. And this opens up a pop-up window. And by default, you have a number of different tracks selected. So here on the right, you can see all the different tracks that are currently enabled. And these are also divided up into tabs. So the tab we're currently on is the region image, so the most detailed view. But we also got a tab for the overview image, so the second diagram of that page. And we also got a tab for the chromosome view, so the very first diagram on this page. And what I'm going to do is I'm going to go back to the configure region image, and I will show you how to add a track. So on the left-hand side, we have loads of different tracks divided up into different groups. So we've got genes and transcripts tracks. We've got variation tracks, regulation tracks. We also have these numbers next to each category. So that simply means, the first number simply means the number of tracks that are currently enabled and the number of total tracks. So for example, for the sequence and assembly category, we've got 3 tracks enabled out of a total of 18 tracks. So I want to add a UniProt track to my region view. So because I know which track I want, I can type it into the search bar on the top of the pop-up window, UniProt. Or you can also just search it in one of the categories here. So as I typed it in, it opened up a new page on the right-hand side here, so search result. And here you can see all the different tracks that have the word UniProt in them. I'm interested in short variants, so I will click the UniProt short variants option here. And when I click this, this box, it will open a window. And here I can select the style of the track that I want. So you can choose to have a normal track, or you can also choose to have a collapsed track, so a bit smaller, and also expanded, so with a bit more detail. I just want the normal track, so I will click that. And that automatically saves it. So once you're happy with your tracks, you can click here on this save and continue, on this check mark on the top right-hand side. Or again, you can just click on an empty space outside the window. So this updates the region and detail view, so the most detailed view. And you know it's still loading when it's kind of still a bit white. So it's finished loading now. So now that we added the UniProt track, we should be able to see it in here. So I'm just searching for that now. And there it is, UniProt variants. And you can, if you've forgotten what a track represents, you can always click on it. And there on the I symbol, you'll always find more information about the specific tracks that you selected. You can also change the order of your tracks. So if you wanted your UniProt tracks to be closer to your genes because you want to compare some data, you can drag and drop it up and down as well. If you wanted to remove a track, you can click on the track and simply click on the X symbol, which disables the track. So now the UniProt track is gone again. And if you ever wanted to reset the whole, configuration, so if you wanted the default tracks and not any extra ones, you can click on this symbol here, which says reset configuration on the top of the diagram. Okay, so this was the regions in detail view. It's quite a short module, but I will go back to the presentation now, and then I'll let you go through the exercises, which are exploring genomic regions. And for this, I'll give you 20 minutes. And again, there are more exercises than you have time for, so just select the ones that you want to do. All right, so it's 14:55 now. Before I move on to the next module, does anyone have any questions? Cool. If you do, just type them into the Zoom chat box or in the living document. Okay, so I'll talk about genes and transcripts in this module. And here I will explain the gene view in more detail and how genes are annotated in Ensembl. I will talk about I will talk about transcripts and how to select the best one, and how Ensembl stable IDs are structured, and I'll also talk about the importance of gene ontology on Ensembl. And finally, I'll do the demonstration, and you will have time to do some exercises. So this is the region in detail view. Here you can see the different transcripts of a gene. So remember, on Ensembl, a gene is defined as a collection of transcripts. So it is normal for many alternative transcripts to be transcribed from a single locus, which is the case for these transcripts here. And each transcript can be expressed differently and can serve a different function. The different types of transcripts are color-coded, and they are in gray, gold, red, and blue. So the gray one stands for non-coding genes, which do not produce proteins, for example, RNA genes. The red one, the red transcripts, represent protein coding genes, which are transcripts that are translated and produce proteins. So that's the red ones here. We also have protein coding genes that are non-coding transcripts and are not translated. So for example, this can be retained intron transcripts or nonsense mediated decay. These can be fragmented and can be missing a translation start site, be faulty or degraded before they can be translated. So these are in blue. And we also have the golden or merged transcript. And this represents transcripts with high confidence in the annotation. So these golden transcripts can be both coding and non-coding. And I will talk more about these golden transcripts in a later slide. Let's look at the individual features of a transcript. So I've mentioned earlier that a transcript is made up of these symbols, boxes and lines. And here you can see the lines, not very well, but you can see the lines and the boxes. So the boxes represent exons, and we've got two types here. We've got empty boxes. So these represent non-coding exons, but we also have filled boxes. So we have one here, and we have here another one, and those represent coding exons. And the lines, there are two different shapes that you might be able to see. You will be able to see them for sure during the demonstration. But here you can see a line that is in a V shape. And these are introns. These represent introns that are located on the reverse strand. And we also have lines that are A-shaped. And these represent introns that are located on the forward strand. If you forget what the different color codes mean or what the different symbols mean, you can always hover over the symbols, the elements, and it will show you what the element is. And there is also documentation on Ensembl if you forget the different codes. So in Ensembl, there are two independent annotation methods that are used, and both rely on valid biological experimental data. And we have the Ensembl automatic annotation pipeline, which is applied to all vertebrate species on Ensembl. But we also have the Havana manual annotation pipeline, which is done by a group of annotators, so people. And this is only applied to human, This is only applied to human, mouse, and partially rat and zebrafish. So the automatic annotation method is a gene prediction method and is based on real biological experimental data. So we take known data, for example, transcripts, proteins, cDNA or RNA-seq, which are then mapped to the genome. And on Ensembl, gene annotation is only mapped when there is experimental evidence that a gene is transcribed. So this automatic annotation process can take up to 2 weeks to annotate a single genome. And the manual annotation method, which is done by a person, a human annotator, can take several years for a single genome to be done. And this is because the manual annotator follows along the genome by hand, basically. This is just a list of annotation sources that are used in the Ensembl annotation pipelines. So we retrieve nucleotide data such as cDNA and RNA-seq data that is submitted to the International Nucleotide Sequencing Database Collaboration, so the INSDC. And this is a collaboration of the European Nucleotide Archive, ENA, NCBI's GenBank, and the DNA Data Bank of Japan, so DDBJ. Proteomics data we retrieve from SwissProt and Tremble. And if there is not enough experimental data for a lesser studied organism available, we also perform annotations based on gene prediction by sequence similarity of a closely related organism. And the manual annotation, manual gene annotation pipeline is done by a person on a case-by-case basis by looking at specific data such as RNA-seq, transcript, structure data, that has been published. So for a manual annotation pipeline, there is a wider range of evidence available to the annotator. But again, this takes much more time, and therefore we only do it for mouse, human, and partially zebrafish and rat. As you might have guessed, the manual annotation process is much more laborious as the person, the annotator, moves along the gene to map published experimental data using different databases. And this can take up to half a day to annotate a single gene. And manual annotation, therefore, is more accurate for difficult feature annotations such as splice sites, but then again, it also takes much longer time. In the Ensembl Genomes browser, the annotation process is a bit different. So we don't do the gene annotation ourselves, but we rely on research groups and communities to keep the annotations updated and to submit this information to publicly available databases. The annotation process by these research groups can be quite similar to the Ensembl annotation, so they can use automatic annotation pipelines by looking at known features. They can do manual annotations as well to correct these automatic annotations, or they can perform homology annotation and find apparent open reading frames in the sequence. So this slide just shows a number of the different species that are available on Ensembl Genomes and the different institutes that have produced the annotation data. So for example, for the common weed, the annotation data was provided by the Earlham Institute, and they used an automatic and a homology annotation pipeline. You can always find out how a genome was annotated by visiting the More Information and Statistics page on Ensembl. So now that you know about the Ensembl annotation methods, if you remember, I mentioned the golden transcript in the first slide of the module. So golden transcripts are just transcripts that have been annotated identically by two different independent annotation methods. So in Ensembl, it would be the case of the automatic annotation pipeline and the manual annotation pipeline. This means that the features of the transcript was annotated the exact same in both methods, the same location, same intron exon set, same length. So that means there is a higher confidence in the function and quality of the transcript. And there is also the GenCode project, which centers around the Ensembl human and mouse gene set. It is a merged gene set of the Ensembl automatically annotated and the Havana manually annotated genes in human and mouse only. And this is the default gene set used by many different projects, such as Nomad, the 100,000 Genomes Project by Genomics England, 1,000 Genomes Project, and so on. And elaborating on transcripts, I want to introduce you to the two canonical transcripts as well. And these are single representative transcripts identified at every locus in every species. So these are transcripts that are most highly conserved and expressed in concordant with other transcript scoring algorithms and UniProt canonical isoforms. So they have the longest coding sequence, the largest number of clinically imported variants, and are complete sequences. And finally, another project that is important to know about is the main select project. So main stands for matched annotation from the NCBI and EMBL-EBI. And it is another measure of the quality of a transcript. So NCBI has its own gene annotation pipeline, which is known as the RefSeq. And main select transcripts are 100% identical in both RefSeq and Ensembl. So the main select transcript is agreed by both databases to be the most biologically relevant and representative for the gene. And finally, when available, the Ensembl canonical transcript is the same as the main select transcript. Okay, so I'll talk about the structure of the Ensembl IDs now. All features on Ensembl, so all elements, transcripts, genes, exons, have a stable ID assigned to it. And this is composed of an ENS prefix, a single letter representing the feature type. So for example, gene, transcript, exon, peptide, and an 11-digit unique number and a version after the dot. Stable IDs do not change between releases. And for non-human species, there's also a suffix added. So for example, for mus musculus, we've got MUS in between the ENS part and the feature type. Or for zebrafish, it would be DAR as a suffix. There is a complete list of these suffixes on this website. So if you open the presentation, all these links are hyperlinked, which will send you straight to the documentation page. And finally, I want to make you aware of gene ontology terms, which are also known as GO terms. Just like in any language, there can be multiple terms for the same biological function of a gene. So for example, one person might call it innate immunity, while another person would call it non-specific immunity. And to tackle this and have a more standardized way of describing functions, the scientific community came up with Go terms to make sure that we are indeed talking about the same function. So Go terms are also meant to be more easily searchable because they have their own unique accession numbers, name, type, and definition as well. And these Go terms are structured in a hierarchy. So a Go term can have a parent term, which describes the wider function of the feature, but it can, oh, sorry, my mouse is playing up. Apologies about that. A Go term is structured in hierarchies. So it can have a parent term describing the wider function of the feature, but it can also have child terms, which describe more detailed functions. So each Go term, so each box in this picture, will have its own unique ID, its own name, type, and definition assigned to it. And if you are interested in learning more about the Ensembl gene annotation system in more detail, I highly recommend this paper. Again, this is hyperlinked in the presentation slides. And with that, I will move on to the demonstration. So we are going to look at the UQ-CRQ gene, and we're going to find out more information about the gene itself and its transcripts. So I'm going to copy this gene name and paste that into the chat box. And with that, I will open our Ensembl tab on the browser. We're still in the region in detail overview, so I'm going to go back to the home page by clicking the logo on the top left. And we're going to enter the gene into the species-specific search box. So I've got it copied already, so I'm just going to paste the gene. And we are interested in humans, so I will select human in the drop-down menu and click go. So this time, because we didn't give a specific location in the genome, the UQ-CRQ search term can apply to a lot of different results. So we are interested in the gene, but what you'll also find in the results are the different transcripts that it transcribes to. If you want to filter the results page down to genes only, you can do that on the left-hand side here. So there is a section that says restrict category to. And because we're only interested in the gene, I'm going to select the gene. So I'll click that, which loads this results page again. Giving us all the genes that have the letters UQ-CRQ in it. So there are two here. I know I'm interested in this one because this is the gene name that we entered there. So I would click the first result. And clicking that first result sends us straight to the gene tab. So before, because we entered a location, coordinates in the search tab, we were sent to the location tab. But this time we are interested in the gene, so we were sent straight to the gene tab. And by default, you get sent to the summary page of this gene tab. So here we can find all kinds of information about the gene itself. There is descriptions, there are gene synonyms, the location of the gene. We've got the different transcripts of the gene. So if you click this button called Show Transcript Table, it will open up a table with the different transcripts that are transcribed from this gene. I will look into these in more detail a bit later. For now, I will close it and show you the rest of the page. So here we've got a summary section with the name of the gene, some descriptions from other databases, also the Ensembl version, and where you can find the gene in other assemblies of the human genome. So similar to before in the region and detail view, if you scroll down, you have a diagram here, which shows exactly that, the region and detail. So here we have the usual tracks. We've got the genes track, the context track, which is the genome assembly track, and also regulatory tracks. So these are all tracks that are already enabled by default. Again, this is interactive, so you can move these tracks around if you want. You can also click on the different features to learn more detail about them. So so that's the summary view, summary display of the gene tab. On the left here, you can see, just like in the location tab, we have all sorts of different displays again. So we are on the summary tab at the moment, but we can also view the gene sequence. We can look into the gene ontology, so the function of the gene, and also some links to the regulation and the comparative genomics data that is available on Ensembl. For now, I will click on the sequence display here on the left. So I will click that link. This opens up, still in the gene tab, this opens up a new page. And this is just the sequence of the gene in FASTA format. And we know it's in FASTA format because it follows this particular structure. So it starts off with a greater than sign, and then with the name of the sequence and a description. Sometimes a description, sometimes there isn't a description. In this case, we've got the assembly, the genome assembly name, and also the coordinates, and also the orientation. So a 1 represents forward strand, and if there was a -1, it would be on the reverse strand. So above the FASTA sequence, above the FASTA file, you will see this box. This is the legend of what the different colors mean in the sequence. So the UQCRQ exons are colored in red font, and all exons in general, so also exons that are not the UQCRQ gene, are highlighted in this peach color. You can choose to download the entire sequence. So there is a download sequence button just above the legend. You can click that. Oh, well, this is not good. This isn't supposed to happen. I will report this straight to our colleagues, but you should be able to see options of what format you want to download your sequence in. So you can download your sequence in the FASTA format. That would be just plain text, just the name and the sequence, or you can also download it in RTF, rich term format, RTF format, rich text format, that's it. And so that would be with the annotation, so with the coloring and also with a legend. Actually, I'm just gonna note down to report it to our colleagues there. Okay. So you can download the sequence, but you can also blast the sequence. So Ben will talk more about comparative genomics data on Ensembl later today. But basically, you can either choose to blast the entire sequence, or you can select, highlight a sequence, and blast just that region, that highlighted region. Okay, so this is the sequence display. And we've also got the gene ontology display, so the functions of the gene. I will click on GO biological process, which will open up a new page, still in the gene tab, but with information of the gene, gene function. So here you can see a list of Go terms. So this is how a Go ID looks like. And this is a list of all the Go terms that are associated with the UQCRQ gene. But as you can see, there are multiple entries for the gene. And that is because a Go term is actually associated with a transcript rather than the gene itself. So each Go term has an associated transcript ID with it. You can click on the different links to learn more about the transcripts as well, or you can filter it down by typing your Go term of interest in here, or even just a term, a word. And you can download the table by clicking on this Excel sheet symbol on the top right-hand side of the table. So those are these are the functions of the gene, but you can also look at gene expression of this gene. So for that, I'm going to go to the gene expression display again on the left, which is a bit further down. I will click the gene expression link. And again, it opens up a new page. And this page shows data from the Expression Atlas database. So not data that Ensembl itself creates, but rather data that is retrieved from a different database. So here you can filter down what you're interested in. So you can, for example, on the right-hand side here, we have a heat map. And this heat map shows the different projects and tissues that were studied of this gene. You can filter it down, which opens a new pop-up by looking at particular tissues that you are interested in. I'm going to close this. You can also download this entire heat map. And when you click these links, it will send you straight to the projects that was done on here. And if you scroll down to the bottom of the heat map, you will also see a legend. So you'll see what the P and the T stands for in the different colors. You can also choose to select a particular sex. So right now it's male. We can also select female. You can hover over the different organs to see the expression there. And you can also choose to download the table content of this entire gene expression page. So now that we've talked about the gene, the different gene displays, I'll show you the transcript table in a bit more detail. So this transcript table is available in all of the displays. So they're all the same, whether I'm on the gene expression display or the summary display, this table remains static. And as mentioned before, this table has lists all the different transcripts that are transcribed from the UQ-CRQ gene. So we've got the transcript ID, we've got the name here, the length of the sequence, the length of the protein. We've got the biotypes. So remember in my first slide, I was talking about these 4 different colors of the different types of transcripts. So if you hover over the words, it explains it again what type of transcript it is. And then you've also got links to external databases here and these different flags. So these are the different measurements of confidence in the transcript annotation. So I've mentioned earlier during the presentation, the main select. We've also got Ensembl, canonical, GenCode, and so on. You can, again, just hover over the different flags with your mouse, and this will show you a description of the different flags. So now let's click on transcript. So I will click on this golden transcript here, the very first one. And if you look to the top of the page, you'll see that a new tab has opened, which is called the transcript tab. We're no longer on the gene tab, we're now on transcript tab. So we're looking specifically at this transcript here. And when you click that link, it opens up the summary page again, just like in the gene tab. That would be your default landing page. And if you scroll down, you'll see a summary of this transcript. So like before, we've got these boxes and lines, and the boxes represent the exons, the lines represent the introns, and you've got some more information and summary statistics down here. So here, for example, you can see the annotation method of this particular transcript. So similar to before, it has a number of different displays here on the left-hand side. So if you click on the cDNA link, it opens a new page, which shows the cDNA display. So it's the matured splice transcript. Scrolling down, you will have this colorful image, which is the sequence of this transcript. It's made up of 3 different rows. So in the first row, this row here, we've got the actual cDNA, which includes the UTR. We have, again, just like before, a legend just above the sequence. So if you're unsure about a color, you can refer to the legend here. The second row is the coding sequence, or CDS, which starts with an ATG. So if you look here, it starts with an A in the first line, TG there in the second line. And then the third row is just the amino acid sequence starting with a methionine. So ATG methionine. If you don't remember what a section is about, there are always, you can always click these question marks, and that will lead you to the documentation page. And also, similar to the FASTA sequence we saw earlier, you can choose to blast the whole sequence, or you can highlight the sequence and just blast that region. And finally, you can also look at external databases that mention this transcript. So scrolling up to the top of the page again, we can see an external references category here on the left-hand side in displays, in the displays section. And here you can, if you click on general identifiers, it'll open a new page, and it will show you all the different links and lead you to the different databases of this transcript that have mentioned this transcript. You can also view protein information of this particular transcript. So again, in the transcript-based displays, we can click on protein summary. And this just includes information of the different domains and motifs encoded in the protein. So we have these different colors, again, with the legend above, with the legend below. And similar to the regions in detail view, you can configure this image and add different tracks to it and change the order. Okay, so that brings me to the end of my demonstration. I will go back to the presentation now. And you will now have time for some exercises. So we'll be working on the genes and transcripts tab in the training page. If you have any questions, let me know. We'll try to do these exercises for 20 minutes, and then I will hand over to Ben to cover the last module, which is comparative genomics. Okay, so it's 3:55 now. Does anyone have any questions before I hand over to Ben? All right, so this will be the last module of the day. Go ahead.
Thanks, Louise. So just to wrap up everything that we've covered today. So Louise took us through an introduction to Ensembl. We looked at the region in detail pages to explore genomic locations. And then we thought about how Ensembl takes those genome assemblies and annotates them with genes and transcripts, and how we can explore the genes and transcripts through the Ensembl genome browser. What I want to do in this final module for today is talk to you about the comparative genomics data that we have in Ensembl. So I'm aware that many of you are sort of studying, obviously DKFZ is an Institute of Cancer Research. So I think many of you are studying human and cancer in particular, but I think it's quite important to still have a brief look at the comparative genomics so that we can understand links between model organisms and ortholog and paralog relationships and those sorts of things. So we're not going to spend too much time, because I don't think it's the focus of many of your areas of research, but I think it's quite a nice module to think about in terms of looking at links to model organisms for your areas of research. So comparative genomics is very useful. Well, sorry. First of all, I wanted to think about what we're going to talk about in this module. So first of all, we're going to think about the applications of comparative genomics and the different species that we have in Ensembl and how we perform those analyses. Then we're going to think about the different types of analyses that we perform. So the first thing that we're going to do is we're going to look at gene trees and homology predictions. So these two areas really cover the comparative genomics analyses that are gene-centric. So where we compare the genes and the transcripts that have been annotated from one species to another. And then in the second part of this module, we're going to look at whole genome alignments. So this is where we forget about the genes and transcripts for a moment, but then look at the alignments of the genome sequences. So the sequence centric analyses. So comparative genomics is really useful because it can help us to understand vertebrate evolution if we're studying evolutionary genomics. And it can also help us to look at differences between species at the genome level. More important for us in terms of looking at model organisms, I think comparative genomics is most useful in being able to understand gene function based on homology between related species. And we can also use comparative genomics to look at the distribution of highly conserved regions across the genome as well that might be functionally important. So all of the comparative genomics analyses that we do in Ensembl are performed within each taxa. So first of all, the Ensembl comparative genomics is performed across all of the vertebrates that we have in the Ensembl genome browser. And then we perform the same analyses on each of the divisions of the Ensembl genome species. So those species, those non-vertebrate species. So for example, the metazoan species in Ensembl metazoa. Then we also perform what we call pan-taxonomic comparer. And this is where we take representative species from each of the divisions in Ensembl and Ensembl genomes. So we take some vertebrates, we take some non-vertebrate metazoan species, we take fungi, some plants, some protists, and bacteria, and we perform all of these analyses on these species to be able to look at the whole tree of life in our analyses. So you can find all of the analyses and the results and the data for our comparative genomics analyses in the browser. So this is an example of a screenshot of the gene tab for EMBL-EBI, so the vector for malaria, the mosquito vector for malaria. And you can see that we have this section here called metazoan comparer. And you can obviously see that this is where you're going to find the analysis of all of the metazoan species against one another. And then we have the section for pan-taxonomic comparer, where you can find the same analyses, but just across all the divisions in Ensembl and Ensembl genomes. So one of the first types of analyses that we produce is called our gene trees. So this is where we take a single protein from each gene that's annotated in every species in Ensembl. And then we perform a process of clustering, blasts, alignment, and other multiple alignments. And then we reconcile that against the species tree to produce our gene trees. From the gene trees, we actually make ortholog and paralog predictions. So we make our homology predictions using the gene trees that we produce. So if we're thinking about a tree that we've produced, and we, as I said, we use those to predict the homology relationships. I just have a small subtree here as an example to show you how we calculate the homology relationships. So homologs come in two main categories. So we've got orthologs and paralogs. So orthologs is a pair of genes that have emerged through a speciation event. So they're two genes in two different species that have a shared common ancestor. So for example, we have C1, which is a gene in chimpanzee, and it is linked to C2 through the speciation event. Sorry, I've misspoken there. So we have C1, which is a gene in chimpanzee, and it's related as an ortholog to H1 through this speciation event, which is in this blue node here. So you can see that there is obviously a common ancestor between chimpanzee and human, and then where this speciation event occurred, this gene in the modern chimpanzee is related to the gene in modern human. The other type of homology relationship that we can think about are the paralogs. So these are genes that have emerged through a duplication event. So these are two genes within the same species where the gene has been duplicated within the genome, and they've obviously diverged over time to have maybe similar or different functions. So this is an example of H1 and H2. So you can see H1 is a gene in modern humans, and if you go back through the tree, you can see that H1 and H2 are linked with this duplication node here. So this is a red node represented here. So obviously in the common ancestor, there was a duplication of this gene that resulted in H1 in modern humans, and then the other duplicate formed H2 in modern humans. The paralogs and the orthologs can be related to each other as well in terms of the number of genes involved in each relationship. So we can have genes involved in simple relationships, so like a one-to-one relationship, where you have one gene in the chimpanzee that's orthologous with one gene in the human. So this is called a one-to-one relationship. But then you can also have one-to-many relationships. And this would be an example of the mouse gene here that is orthologous with the two human genes. So what you can see here is if you go back to the top of the tree, there was a speciation event where the rodent and the primate lineages diverged from one another. And in the rodent lineage, this gene just persisted to modern mouse. Whereas in the primate lineage, there was this duplication event. So that means that there's now two genes in human that are orthologous with the single gene in mouse. So this is a one-to-many relationship. And of course, you can also have many-to-many relationships. And this would have happened if there was a duplication event somewhere in the rodent lineage as well. So what we're going to do in our demo is have a look at the human BRCA2 gene. And we're going to find the homologs of the BRCA2 gene in a number of different species. So from in your internet browser, you can type www.ensembl.org, and that will take you to the Ensembl homepage. I think we're still having problems with the speed of the website today. Here we go. So this is the homepage. Obviously, Louise introduced you to the homepage earlier on today. And we've also used the search box to search for genes. So we're going to do the same thing as we did in the last module. We just, into the search box, we can type our gene name of interest. So in this case, it's BRCA2. I can put this in the chat box so that everyone can copy the gene name if they need to follow along in the demo. And then when you're happy, you can click go. So just as many of you are doing, I've seen many of you doing this in the exercises already. I think you've been using the mirrors, and I've suggested it in the chat box as well to a few of you. So I'm going to use one of the mirrors to try and speed up the internet, the website. So I'm going to use uswest.ensembl.org. I'll put the link into the chat box as well, so you can also navigate to US West. So this is often a problem that happens if one of our servers is particularly busy in either Europe or the US. So sometimes it's worth just flicking between the different servers if there's a temporary issue. So I'm going to try this again. So from the US site, I'm going to type BRCA2 in the search box and click go. Okay, still quite slow, but it is working, so that's good. So from the results page, we can click on the link for the human gene to take us to the gene tab. So many of you would have seen the gene tab already. And you can see the link over in the menu on the left-hand side. There's this whole section called Comparative Genomics. So the first thing that I want to do is click on Gene Tree. So if we click on Gene Tree, you'll be able to see the gene tree that has been built from the proteins and using the clustering and the blast alignment that I was describing to you earlier. So if we look at the tree, you can see that you have the human BRCA2 gene here highlighted in red species. So obviously in the branches close to human, you have the other primates. So you've got, well, the other great apes. So you've got gorilla, 2 chimpanzees, orangutan, and then obviously some of the other primates further away. What you might notice is that there are these gray triangles on the tree, and these are just collapsed subtrees that help to help to display the tree in one page. So as you can see, for example, under this gray triangle here, we have 24 different primate and rodent orthologs that have just been condensed. So if you want to see the full tree, you can click on these triangles individually. So if you click on one of the triangles, in the pop-up window, you'll have this option that says expand this subtree. So you can see that section of the tree is now being expanded to show all of the species that fall in that part of the tree. So you can see here all of the primates and rodents that were there. Just as I had in the cartoon, in the presentation, you can see that the nodes are color coordinated. So you have the blue nodes, which are the speciation nodes, and then you have the red nodes, which are duplication nodes. So you can see here, this is a duplication node because golden hamster has 2 orthologs of the BRCA2 gene here. Over on the right-hand side, you can see these green bars. So these green bars are representative of the protein alignments that the gene tree is based upon. So you can see, if you go to the very bottom, what the green and the white sections mean. So the green is where we have an alignment between the protein, the human protein, and the other proteins in the tree. And then a white section is where there's a gap in the alignment between the different proteins. There are some proteins that have this dark green coloring, and this would correspond where you have these collapsed trees. So these are what we call collapsed alignments. So this obviously is one alignment that's meant to represent all of these 3 New World monkeys that's hidden underneath this section of the tree, for example. And here, for example, we have 52 bony fish, and again we have this collapsed alignment. Represented by dark green is between 66 and 100% aligned. The light green is between 33 and 66% aligned. And then the white is between 0 and 33% aligned. Good. So obviously you can see the orthologs within this tree. But one of the other things that you can do is you can view a table of the orthologs. So if we click on ortholog, the menu on the left-hand side, this will take you to a page that shows us all of the orthologs of the human BRCA2 gene. So at the top, we have this summary table, and you can see at the moment we are showing all of the species in Ensembl and their orthologs that are there. So there are 165 species with a 1-to-1 ortholog. There are 5 species with a 1-to-many ortholog, and then there are 30 species that do not have a predicted BRCA2 ortholog. So if we scroll down, we can begin seeing a full table with all of these species. You can see that, for example, the alpaca has a 1-to-1 ortholog with the human BRCA2 gene. Obviously, this table is quite big because of the number of species we have. So you can actually just use this filter in the top right-hand corner to begin searching for your species of interest. So if you're using mouse as a model organism, you can begin typing mouse. And now you can see all of the species that have mouse in the name. So we have obviously Algerian mouse, but then also species like mouse lemur. So it's not obviously done on taxonomy, it's just done on the name, the species name. But if you scroll down, you can see that we have mouse mus musculus. So this is the reference mouse genome assembly. And we have a 1-to-1 predicted ortholog to the human BRCA2 gene. So again, the mouse gene name is BRCA2, and then this is the ID of the mouse gene. So if you want to find out more information, you can click on the link here to go to the gene tab for the mouse BRCA2 gene. In the table, you can also see that you've got some scores to tell you about the percentage of the alignment between the 2 genes in human and mouse. So we have the target and the query percentage ID, and then we have a gene order conservation score and a whole genome alignment coverage score as well. What I wanted to point out to you is in this table, you can click on the link called View Sequence Alignments. So if you click on that, you'll get a small pop-up window that allows you to either view the protein alignment between the human and the mouse BRCA2 genes, or the cDNA alignment. So if you click, for example, on View cDNA Alignment, it will take you to a page with the cluster W alignment between the human and the mouse genes. So you can see here, this is the cDNA sequence of the human BRCA2 gene, and this is the cDNA sequence of the mouse BRCA2 gene, and you've got the cluster W alignment with the scores underneath as well. And you can download this alignment if you want to, using the download homology button at the top there. Good. Okay, so that brings me to the end of the gene trees and ortholog analyses that I wanted to show you. What I want to do now is just go back to the presentation and quickly show you some of the alignment data that we have before our exercises. So, as I said before, in the previous half of this module, we were looking at the gene-centric data. So we were looking at how Ensembl compares the genes from one species to another. In this second section, we want to think about how we can compare the genome sequences from one species to another. And we do that through doing whole genome alignments. So whole genome alignments are useful to identify highly conserved regions. So these highly conserved regions are likely to have evolved slowly, and they're therefore likely to be functional. But it can include both coding and non-coding sequences. So we have two main types of whole genome alignments. We have the pairwise and the multiple whole genome alignments. So the pairwise whole genome alignments, as the name suggests, are done between two species against one another at a time. So we take, for example, human against macaque, or rat against mouse, or zebrafish against the Amazon molly fish. So, for example, we take human and we have a pairwise alignment against each of the other species in Ensembl individually. And then for model organisms, we also do alignments against related species. And then the agricultural animals we do against one another as well. The second type of whole genome alignments is the multiple whole genome alignments. So this is where we take species sets, so predetermined species sets, and we perform the whole genome alignments across all of these species in one go. So for example, we have a pipeline called the EPO pipeline, the Enredo Pecan Orpheus analysis. And this is where we take closely related species such as the 12 primates in Ensembl, and we perform whole genome alignments across all of those species. We also have what we call the EPO extended analysis, and this is for larger species sets that have fragmented assemblies as well, so lower quality genome assemblies. Then we have the Mercator Pecan analysis, and we use this to perform the whole genome alignments against the amniotes, so the mammals and the birds. So the EPO pipeline that I described above is really good for performing whole genome alignments, multiple whole genome alignments for closely related species. But the Mercator Pecan analysis is a lot more tolerant of distantly related species and differences in the genome sequences. So we use this for looking at the mammals and the birds, so the amniotes. And then we also have this cactus alignment software that we use to look at all of the different mouse strains that we have in Ensembl as well. So we have a number of mouse strains that you can look at. So what we want to do in this demo is have a look at the human genomic region that contains the HOXD cluster of genes. So it's a region on chromosome 2 that contains a number of genes that are involved in embryonic development and limb formation. So they're very, very important genes that are highly conserved across a number of mammals. So we're going to look at the genomic region that contains this HOXD cluster, and we're going to see how we can look at the alignments across species. So again, if we go back to the Ensembl genome browser, I'm just going to go back to the home page by clicking on the logo in the top left-hand corner. And just as Louise showed you before, we want to search for a genomic location. So from the search box, I'm going to click on human, and then I'm going to search for my region of interest. So I'm going to put the coordinates in the chat box for you as well, but I can type them out here. So the chromosome number is 2, then we have a colon, and then our start coordinate is 176.202.000. Sorry, 176.087.000, and then a hyphen, and then our end coordinate is 176.202.000. So I'll put that in the chat box for you to copy and paste and follow along with me. So once you've entered the coordinates, you can click on Go. And that will take you to the location tab. So just as you saw earlier with Louise, you have this location tab that shows you the genomic location with the tracks all aligned to the genome sequence in that region. So you can see at the bottom, the page is just loading. But what we can do while the page even loads is we can begin looking at the tracks that are available that show us the alignment data. So I'm going to click on Configure this page, the blue button in the menu on the left-hand side. And in the pop-up window, you can see the section over on the left-hand side called Comparative Genomics. So you click on the title, Comparative Genomics, in the menu on the left in the subcategories, and it will show you all of the comparative genomics tracks that we have available to us. So at the moment, you can see that we have 2 tracks turned on, one called Age of Base, and then the other called Constrained Elements. So the constrained elements are the regions of the genome that when we perform an alignment across the 91 eutherian mammals, so these are constrained elements for the 91 eutherian mammals. So when the whole genome alignment has been performed across these 91 mammals in Ensembl, we've identified those regions of the genome that are highly conserved across all those species. So what we can do is we can turn on the track above for the conservation score. So that will tell us, so what we do is we perform an alignment, as I said, and for each base, we give a score across the 91 species about how well conserved that particular position is. So if it's highly conserved, it has a high score, and if it's poorly conserved across those species, it will have a lower score or a negative score. And then we look for regions of high positive score, and we label those as constrained elements. Further down, you can see we have a section for all of the pairwise genome alignments. You can see here we have the BLASTZ/LASTZ alignments, which is the pairwise alignments. So this is always going to be human because that's the species that we search for against each of these individual species. So I'm just going to select 2 or 3 as an example here. So I'm going to search for maybe chimpanzee as a closely related species. And then I'll also choose cow because it's a mammal, but more distantly related. And I'll choose normal as the format. And then maybe I will choose I'm trying to find like a more distantly related species. How about the common wall lizard? Here we go. Okay, so once you're happy with your tracks that you've added, you can click on the tick in the top right-hand corner. And you can see at the bottom, my page is just loading. So you can see we have these tracks that are now turned on. So we have these two tracks at the top here. So we've got the 91-way GURP elements and the 91-way GURP scores. So this is the scores and the constrained elements that I was telling you about just now. So as I said, we perform a multiple whole genome alignment across the 91 species eutherian mammals in Ensembl. And for each base, we give a score of how well conserved that position is across those 91 species. So we get this scale on the x-axis, on the y-axis between from positive score to negative score. So you can see there are regions of negative score here which are poorly conserved. And then you can see that there are regions of positive score, like we can see here, which is where we have these constrained elements that are annotated. So where there's a block of positive score altogether, then we annotate that as a constrained element. You can see here there's this whole stretch of negative score here, which is why there's this no constrained elements annotated here. If we scroll down, what you can actually see as well is that this is the HOXD cluster of all the different HOXD genes. So we've got HOXD13, HOXD12, 11, 10, 9, 8, etc. And you can actually see that the coding sequences of these HOXD genes actually very closely overlap with the constrained elements. So wherever you see these constrained elements, it's likely that these are coding sequences in this HOXD. If we scroll all the way to the bottom, you can see that we have our alignments of the three species that we picked in pink. So we've got obviously human against chimpanzee, human against the common wall lizard, and human against cow. So the pink bar represents alignment between the two species. So as you might expect, human and chimpanzee are very closely related. So we have this pink block that shows us that there's alignment across this whole region between human and chimpanzee. Between human and cow, you can see that there is some gaps in the alignment, which are represented by these white blocks. And then for the common wall lizard, the most distantly related species we picked, you can see that there is some alignment with these pink blocks, but then there's also big gaps in the alignment as well. Good. So the final thing that I want to show you, if you scroll back up to the top of the page, is how you can actually view the alignments between the species in Ensembl. So there's a section here in the menu on the left-hand side called Comparative Genomics. So you can explore all of these different options in your own time, but the one that I want to look at the moment is the alignments text. So if you click on alignments text, this is where you can actually view the alignment between human and then any species that you select. So the first thing you have to do is click on this blue button that says select an alignment. And from here, you'll have a species selector window. So this will allow you to select your species of interest. So you've got two options here. You can either begin typing your species of interest into the search bar. So you could type mouse, for example, and then choose mouse very simply. And it's added to the species, the selected species over on the right-hand side. I'll get rid of that to show you as well. You can also click through from these options here. So this can help you explore the different alignments that are available. So you can click on the multiple alignments or the pairwise. And then once you click on pairwise, you can begin looking at the different species that are available. So maybe if we look at the rodents and lagomorphs, then the rats and mice, again, you can find the mouse reference this way as well. So then you click on your species and it will add it to the right-hand side. So once you're happy, we'll use mouse as our example here. Once you're happy, you can click on apply. And this will take you to a page that shows you the alignment blocks that have been calculated for this region of the human genome against mouse. So you can see that there are two alignment blocks between the human HOXD cluster, the region that we searched for, and the mouse genome. So there's one on human chromosome. There's one block of 115,000 base pairs that's located on the mouse chromosome 2, and then there's a shorter block of just 500 bases located on mouse chromosome 7. So maybe I would probably guess that this shorter alignment block is maybe an artifactual block, or something that is not so interesting for us. This alignment block here comes across a really large region. So I'm guessing that this is the analogous homologous region in mouse. So we can click on block 1 here in the table, and we can view this alignment block. So at the top, we just have a description here, but then if you scroll down, you can begin to see the alignment at the bottom. So this is just the first row, but you can click on display full alignment, and it will show you the alignment between the human and the mouse genome. So obviously there are gaps in the alignment, as you can see here. And then you can also see further down, there are these red sequences, and these are the coding sequences or the exonic sequences in the human and the mouse genome. So if you scroll back up, again, just like before, you can download the alignment just as you did before, if you want to download it and begin working with it. Good. Okay, so that brings me to the end of the demos. So what I want to do now is give you a chance to have a go at the exercises. So it's well, it'll be 4:30 over for you in Germany. We're due to finish at 5. So what I suggest we do is maybe have 15 minutes to have a go at these exercises, and then we'll come back together in 15 minutes at 4:45, and we'll see if there are any final questions before we finish up for the day. So please have a go at the exercises. They're on the training pages. I haven't opened them up yet, actually, but from the training pages that I'm sure you'll all have open already, you can find the comparative genomics exercises here. So we have a set here, comparative genomics exercises, and we can look at ortholog relationships and alignments as well. Hello, everyone. It's just gone 4:45. So I wanted to bring us all back together to see if there were any final questions before we finished for today. So there is one question in the chat box that came in just a moment ago. So I was planning on answering this together with everyone and then seeing if there are any final questions before we wrapped up. So Ricardo's asked if there's a way to obtain the alignment of all the paralogs of a gene, something that looks like the Wasabi alignment that they've found in the browser. So you can view alignments between paralogs, and I can quickly show you this in the browser. So if we go just to the Ensembl homepage, I'm going to search for a gene called VEGF-A. So from here, from the results, you can click on the link for the gene tab. So if there are paralogs available for a specific gene, like there is for A, you can see this in the comparative genomics section in the menu on the left-hand side. So you can click on paralogs. And you can see here, these are the paralogs of the human VEGF-A gene. So you've got the VEGF-B, VEGF-D, PGF, and VEGF-C as well. So what you can do is you can actually click on these links here for either a region comparison. So looking at the regions of these two genes side by side of the genomic regions, you can look at the protein or the cDNA alignment between the human and then the different paralogs. So hopefully that's what you wanted. You can also download the paralogs. So you can download them, you can download the alignments here, however you want to, in all these different formats. So that might help you get what you need. Hopefully that's what you're after. Good. So what I want to do now is just quickly check to see if there are any final questions. So I'm going to stop sharing my screen at this point. So what I want to, so please let me know if there are any final questions. I've been looking in the living doc and the chat box, and I think all of the questions have been asked. If there are no final questions, then I think it's a good time to stop for today. We've covered quite a lot already. So what's going to happen is we're going to have the same time and the same link for tomorrow. So we'll be starting at 1 p.m. German time, going through to around 5 o'clock. And we're going to cover the variation data and the variant effect predictor. Then we're going to move on and look at regulation data to look at features that regulate gene expression. And then we're going to be finishing off with Louise, who's going to be talking to us about the Biomart data export tool. So hopefully you found today useful, and hopefully it's been a good introduction for some of the advanced topics that we're going to be covering tomorrow as well. If you do want to continue having a go with the exercises and looking through Ensembl this evening, then you're more than welcome to. And if you do have any questions, you can put them into the living doc, and Alina, Louise and I will get to look at those tomorrow morning, and we can put some answers in before the session begins. So hopefully that was useful for you, and we look forward to seeing you all tomorrow.